All right. Welcome to the last session already. We still have three excellent speakers, and after which we will have a closing discussion. So it's my pleasure to first announce Dr. Antonio Garcia-Guerra from the University of Oxford, who will discuss his Mirage system for programmable RNA sensing guide RNAs. And the floor is yours.
Okay. Thank you very much, Oli. So hello, everybody. My name is Antonio Garcia-Guerra. I'm a postdoc at the Carlo Rinaldi Lab and Matthew Wood's lab up in the Hill. And I'm going to talk to you today about programmable microRNA sensing guides for tissue-specific CRISPR activity in vivo. All of this starts with the concept of genetic medicines, and we all know how genetic medicines are going to revolutionize medicine, and that is because they can do something that we couldn't do previously, particularly in diseases. One of them is which we can actually start treating and in some cases cure diseases that we just simply could only manage symptoms before. However, because of the nature of these kind of new therapies, which normally are nucleic acid based, we still have issues, and one of the limitations, one of the biggest ones, is delivery. And that is because nucleic acids are, even though we can stabilize them, their pharmacokinetics are not generally particularly well to accumulate whatever we need them to be. And although there is huge advantages of doing or utilizing different administration routes to bring the therapeutics, whatever we need them, in a more local way, when you actually start doing systemic delivery and other issues, you start to face a problem with delivery, which is a very complex process produced through different barriers that need to be overcome simultaneously. And one of the big issues that you're going to have is the issue of off-target accumulation on bystander organs or particularly cell types. And that is, in some cases, very relevant in the context of gene editors. And that is because at the end of the day, you would not like to have anything dealing with modifications at the genome level unless it's strictly necessary. So just to, as you have noticed, we're going to start talking about CRISPR systems and gene editors. So this is just as a quick recap of what the CRISPR systems are. And basically, they are a proto-immune system found in prokaryotes, in which an RNA-guided nuclease targets invading exogenous DNA, in this case phages, and deactivate them by introducing double-strand breaks. And then this information can be stored in what it is a CRISPR array, which basically directs the nuclease to cut in those specific sequences. And what researchers realized is that that system could actually be repurposed to target different loci within the genome. And that is exactly how it raised the whole point of CRISPR therapeutics at this strategy. However, what you have noticed is that there is, by now, all the attention to CRISPR has evolved into the production of different modalities in which not only this nuclease can actually introduce double-strand breaks, but they can actually modify DNA, and in some cases, and also RNA in different ways. For instance, you can have base editors that modify bases. You can have prime editors that can introduce small pieces of DNA within target DNA sequence. You can actually turn genes on and off using transcriptional regulators. And then you can, on top of that, do that at the RNA level. But the most important thing of all of them is that all the effort has been done on the protein engineering side of things and how to create more complex proteins with more sophisticated modalities. And there's not much attention on the RNA. And one of the interesting parts of bringing attention to the CRISPR RNA or the guide RNA is that all of them share a guide RNA. So you could potentially do a modification in a guide RNA for that nuclease of a specific species, and all the modalities could, in principle, could use the functionalities on those guide RNAs. And this is where I introduced the concept of microRNA activated genome editing, or CRISPR Mirage, in which what we are trying to do in here is try to address the issue of delivery in terms of target accumulation, by which we are basically creating a system that is only active wherever specific signatures of microRNA are present in the body. And we managed to do that by introducing a two-state guide RNA. So the wild-type guide RNA, it just finishes in here, and with like the blue sequence guides the Cas protein to the loci in which it's going to cut. And then it has a constant secondary structure that helps the RNA being recruited in the CRISPR into the Cas9 nuclease. But we have introduced this other hairpin that creates two different states. One constitutively off switch that actually creates no Cas9 activity, And then we have introduced microRNA binding sites that when they recruit the microRNA loaded into an argonate protein, then that promotes a change of conformation that allows the loading of the guide into Cas9 and therefore the activity. In order for do all the screening, all the different conditions and all the different all the different mechanisms for the guide RNAs, we use the stop live reporter. I imagine that you guys, this may actually ring a bell. This is exactly the same fluorescent reporter that Oli presented yesterday. We were actually collaborating with him and he was providing it with us. But just to bring this into a very simple level, what this construct allows us to have is a constitutively expressed red protein, so cells are always red. And if you have productive Cas9 activity on this specific portion on the linker, in 2/3 of the cases, you might reestablish the frame of one of these CGFPs, and therefore your cell will turn green, which basically translates in a very simple thing, which is, and a very simple statement, which is if the microRNA is not present, the cell is red. If the microRNA is present and produces successful CRISPR activity, the cell becomes green. But I've been talking about microRNAs right now, and I just want to take a step back on the issue of what we were talking about, accumulation of target tissues and bystander tissues. So why microRNAs are important for this case. So microRNAs are small non-coding RNAs that the cell uses for post-transcriptional control of gene expression. And they were discovered by Victor Ambros and Gary Ruvkun, which culminated in the Nobel Prize of 2024 in Physiology or Medicine. The interesting part about microRNAs, beyond their abilities to regulate transcripts and their translational repression, degradation, or mRNA deanylation, is the following, and is that microRNAs actually can be used as tissue or cell type markers. And that is because their expression profiles can be unique to specific cells, cell types, or particular tissues. And this is normally something that you can find with the specific signatures of microRNAs, but there might actually also have microRNAs that are specific to a tissue. For instance, you have this very well characterized muscle myocard, like microRNA 206, or then you have another one that is very famous for the liver, which is microRNA 122. And please keep in mind 206 and 122 because we'll talk about them later. You have other ones for brain, but just the point of the matter here is that microRNAs can be used to actually determining which cell type you find, which help us address the issue of off-target accumulation, therefore having CRISPR activity exclusively whatever those microRNAs are. So if you bring then the next point to here, it has been a bit of a discussion that I won't go into details, but we've been talking a lot about a bit of microRNAs, strand displacement and everything. And actually the interesting part is the microRNAs within a cell, they behave a bit differently than when you take them outside. And one of the first key points that we needed to address was the difference between, sorry guys, I'm Spanish, so that's why I have that. It's the difference between microRNA activity and microRNA expression. So the first thing that people think of microRNAs, or when they talk about microRNA profiles, is particularly expression. You have the microRNA present in the cell. And normally what they do, they're stabilized by being loaded into an argonaute. Problem that you have with that is that microRNA expression is not correlated with microRNA activity. And the one that we want is microRNA activity. And what means is a microRNA activity means a microRNA that is available to regulate transcripts that have a microRNA binding site. So what you have over here is a qPCR. These are microRNAs that are under this threshold are actually expressed, and microRNAs that are at this threshold not expressed. So these ones should be present in the cell. But what you can see is that microRNAs that are actually expressed in the cell, like microRNA 10, when you put them in a dual luciferase assay, which you have a vanilla luciferase to normalize, and then a firefly to measure its activity, microRNA 10A cannot regulate firefly. So actually it's not active. So once you start going not for expression, but you start to going for activity, that's when actually you get CRISPR activity. And this is the dual luciferase system, the stop light reporter from Oli. So what we are measuring here is the amount of green cells normalized to the amount of red cells. So when you get green signal, you get activity. And we only get activity in those microRNAs that are actually active. And just for also very important, goes back to what I was saying on argonaute and strand displacement, is that microRNAs within cells exist loaded into argonaute, and actually the interactions that happen at this level are not generally mediated by a normal strand displacement reaction. You actually need to understand the biology of argonauts. And what you can see over here is that when we introduce mismatches in positions that are very well documented in the literature for affecting the Argonaut behavior, that's when we get activity. And what these mismatches are doing is actually promoting release of the binding. So what we are actually is promoting the catalytic process of Argonaut. It comes, binds, opens the hairpin, and then leaves. If you don't introduce these mismatches, then the binding is potentially very stable and therefore produces no downstream activity. So the next question that you may have will be, Antonio, do you know if that is actually microRNA mediated or anything like that? So we actually looked at using antagomeres, so like small antisense oligonucleotides that block microRNA functionality. And what you can see is that when we compare a specific microRNA with its antagomere, then you can see activity, and when we have produced the antagomere of it, there's no activity, and so on for different microRNAs. So we are fairly certain that the microRNA is not seen here. It's actually microRNA mediated. And the other point that you will have in mind is like, okay, this is binding microRNAs. Is that affecting physiology itself? Like other transcripts may be dysregulated because a microRNA should be binding in there. So we looked at microRNA 17, and we took 3 different genes that are known to be regulated by microRNA 17. And what we saw was that actually the expression of those guide RNAs in the cell does not affect the expression levels of that specific transcript, except when you add an antagomere as a control, that when you dysregulate with an antagomere microRNA physiology, you see an upregulation of the transcript. So when we add our MER guides, you see no effect on it. So actually, it's not affecting the microRNAs inside the cell. But if we go back to the concept of microRNAs, you will have noticed that in some cases you may actually have one microRNA that can provide activity that you want, but in other cases you may want more than one. So we took advantage of the fact that the guide RNA for SPK9 is actually a 2-strand system. You have the transactivated RNA and the CRISPR RNA, and we introduced the same hairpin, but just in different positions of each of the strands. And what that actually allowed us is to create a Boolean in which you can actually only get activity if you have both microRNAs. I could go through the graph, but that would take us a little time, but I don't have, and these are all the controlled with antagomeres and different microRNAs. But as you can see, when you have both microRNAs in it, you actually get activity. So we can actually target not only one, but several ones, several if needed. And then we decided, okay, will this be actually helpful in any particular context on a medical case? So we chose DMD. So Duchenne muscular dystrophy is an X-linked neuromuscular disease in which there is muscle deterioration due to the loss of dystrophin expression. And it has a huge number of mutations along in specific positions, which are called hotspots mutation sites. We started to look into a specific set of mutations, the delta 52, which what basically means is that it's as exon 52 has been deleted, and that changes the frame of that specific RNA, creating a stop codon, a crematory stop codon that results in a protein that doesn't work. So what you can do is target Cas9 into the splice sites, acceptor sites, and that can result in two different things. Can result in the exon 53, which contains the stop codon, resulting in the production of the protein, or you can actually introduce an indel that may reframe the gene, therefore, again, resulting in the production of the protein. So what we went here was to use microRNAs that were controlled and that were tissue specific. We used one that is muscle specific, which responds to microRNA 206, and one that is specific to liver, which responds to microRNA 122. So we started first working with human myoblasts, and what you can see here is that the myoblast responds to the MER guide that is muscle responsive, but does not respond to the microRNA that is liver responsive. And we characterized all the different mutations, and you can see that even though we looked at here at the mutations at the RNA level, they also transfer at the protein level. You see here on the myomer restoration of dystrophin, which you don't see in the liver, and this is a small ICC pictures in which you can see the restoration of dystrophin. And we took it a step forward and looked at it into animals. We used 2 viruses for the delivery of one of Cas9 and guides. We injected an MBX52 mice that recapitulate the delta 52 mutation in the case of dystrophin. And what you can see that in muscle tissues like diaphragm and hearts, you can see that the myomer actually restores the production of protein, but when you look at the liver mir, you don't have that much restoration, or in some cases not. But when you look at brain, which is also a dystrophin expressing tissue that does not have microRNA 206, you have positive restoration on the positive control and nothing on the microRNA control. It doesn't have two of this. And here is just a restoration of the dystrophin. So I just wanted to basically show that this actually works in vivo and it could be deployed in a clinical setting. So I just wanted to thank the labs in which I've been, that have helped and collaborated to make this research possible. And I'll be really happy